opportunity to look at different food samples and we will see if they have the presence of salmonella or any other enteric bacteria. So that becomes our today's purpose number one. Second purpose is to differentiate between enteric bacteria. It can be salmonella, it can be proteus, shigella, escherichia. So our second objective is to differentiate between different types of enteric bacteria based on three activities. So first one would be morphological differences. Second one would be based on TSI slant test, which is triple sugar ion slant test. And third one would be urease activity. Coming to the prior knowledge, now we know that salmonella is an ubiquitous bacteria and it plays a significant role in developing risk assessment models and in developing formulations of food products. Now we know that there has been recently many outbreaks from the salmonella and it causes diarrheal diseases like salmonellosis. So it becomes significant to isolate and detect the presence of salmonella from the food samples and from the environment. Coming to the gaps, now you have been provided with the food sample, but we do not know whether it has the presence of salmonella or not. And if it has, we do not know what kind of species is it. Since there are many serovars of salmonella species, so we do not know what kind of it is. Next, coming to the hypothesis. So just as a hint, you will build your hypothesis specific to the food product. So recently the outbreaks in US has been related to the raw and the meat products. So based on the type of food sample that you are dealing with, depending on the processing and the environmental conditions. Next three parts focuses on the differentiation of the two types of strains. So part 2 is based on the morphological differences. So for this, you have been provided two isolates, that is isolate 1, which is S. aureus, provided in yellow tube, and second is the isolate 2, that is Staph epidermidis, which is in red tube. So S. aureus is coagulase positive, which means it is an enterotoxin producing strain, and S. epidermidis, it is a non-enterotoxin producing strain. So for this, what you have to do, you have to first carry out the serial dilution up to 10 raised to power minus 6 and then spread plate it for 10 raised to power minus 5 and minus 6 again on Braid Parker oven plates. So each student for this lab will perform with both the isolates. Next comes the part 3, which is the second test to differentiate the strains, that is coagulase test. So for this test, you will take coagulase plasma tube, which is kept in the refrigerator. That contains 0.5 ml of coagulase plasma. You will add 0.5 ml of isolate 1 and isolate 2. So that is, each student will take two tubes, one for isolate 1 and second for isolate 2. Simply add the isolate and incubate it for over. So this is a pretty simple experiment. Now this is based on the principle that if your strain produces coagulase, it will react with the coagulase reacting factor which is present in the plasma, and it will form a thrombin complex which will convert the fibrinogen to fibrin, and you will see the clotting of the plasma. Part 4 comes the differentiation that is based on the third method, DNA. What is the significance of evaluating hygienic conditions in a dairy processing plant or food processing plant? So this is done with the three primary objectives. First, to evaluate or to verify the effectiveness of the CIP protocols which are being followed in the dairy plant. Next, to determine the frequency of those CIP cycles. Second objective is to determine the presence of foodborne pathogens and their potential source of contamination. Third objective is to evaluate the hygienic design of that production facility and the equipments in that facility. After the why, now comes the question how it is done. So this is done with two considerations. First, zoning concept. So every production facility is divided into 4 zones. First zone, which is the direct product contact surface area. For example, inside of a raw milk silos or inside of any crystallized tank, spray dryer. So that is the example of, which is away from the zone 1 and 2, but still it is in close proximity. Next comes the zone 4, which is the remote area, and it is not in any close contact with the first 3 zones. So this is the background information. For today's lab, session 1 is divided into 3 sections. So first section would be flow germ demonstration, which is we want to know how clean our hands are before and hand washing experiment. So this is a voluntary activity. First, I will demonstrate it with my hands, and if anyone would like to perform with their hands, they are most welcome to come and we will help you to demonstrate that activity. Next comes the section 2 of this session 1, which will be performed in the Davis Dairy Plant. So for section 2, we have identified 3 zones in Davis Dairy Plant for which you have to take these swab samples. So zone 1, you have been provided with a reference sheet. So zone 1 is the inside of a crystallized tank in the Davis Dairy Plant. So for that, you will take surface protein plus, which is this purple stick, and 2 quick swabs, which is this red stick. Next comes the zone 2, which is the outside of the crystallized tank. For that, you have to take 2 quick swab samples with this red one. Next comes the zone 3, which is in the hallway. You can take swab from the walls, from the doors, or from the floor tiles. So for that also, you will take 2 quick swab samples with the red one. And for this activity, 3 groups will go in one go. That is, 6 students will go and they will take the swab samples. And Ratul will be there in the Davis Dairy Plant to help you. Next, how to perform these swab samples with surface protein plus. So for the purple one, first you will label it with your initials and then take out the swab from the tube. First you will moisten it with the HPLC grade water, which is being provided in the tube in your kits. And then you will take the swab sample from inside the crystallized tank. Now how to take the swab sample? So first, you will identify an area from which you want to take the swab sample, and this needs to be in the area of 10:10. For example, you are taking a swab like this. First go in one direction and then in the opposite direction. So that is how you will take the swab sample with this. Then insert back, but do not push this stick inside. You will do this when you will reach lab 057. Because it is a time sensitive test, when you will reach in 057 lab, just press it, wait for 10 minutes, let it incubate. And then
they're like, fill this blue basket with quick wash. And we get out there like, we needed double this. And we're like,
plus
of the ATP.
Right? Now you are taking
Oh gosh.
it back
For the one time
and wait until
that
you
we went and took all of these
After
boxes and like threw them in the fridge.
and after you